Okay, welcome back. This is Issues in Biotechnology, the way we work with life. Again, we're into the second part of the course on applications of biotechnology. And on this section, we are covering forensics. This is comprising trace evidence, DNA-based forensics, and the national debate over public safety versus the right of privacy. So in this lecture, we're going to focus primarily on DNA-based evidence. We will post a lecture by Amy Duhiem, which does an excellent job at looking at the evaluation of trace evidence that comprises then everything from hair analysis to bullet casings and how arson is analyzed, etc. Tonight, however, we're going to focus on DNA-based forensics. So I want to acknowledge the Rhode Island State Crime Lab for their contribution to this series with the trace evidence section. I also want to recognize the Connecticut State Forensic Science Laboratory, and over the years their contribution, particularly Carl Ladd, who's the lead criminalist there, and Mike Adamovich, have been lecturers in this class in the past and have helped me put together some of the material for these lectures. I also want to remind you, whether you're on campus or online for this class, that the Biotechnology Stock Project is due. Recall that at the beginning of the course, you received $100,000, fictitiously, of course, and that you selected and researched 5 biotechnology companies. And those could have been in any of the sectors, whether that is agricultural, pharmaceutical, medical, and invested in those by choosing the stocks, looking up their stock quote and the ticker symbol for the company they represent, and calculating the number of shares that were purchased. At this point in the course, you are asked to submit a report to calculate the losses and gains as have accrued over the time period of this course, and submit your final report online. To whom much is given, much is required. Once you know, you cannot unknow. In much wisdom there's grief, in much knowledge there's pain. Now that you know that you know. That latter line comes from a novel that I wrote that you can find available on the Life EDU website, called The Abandon. And I think it's an appropriate lead-in to what we would uncover tonight about forensics. A lot of people are drawn to this subject because of the popular press, but I want to inform the debate tonight about how it's done and how it's carried out, as well as some of how these investigations are followed through on. So we want to focus primarily on DNA-based evidence in forensics. But before I get to that, a little poll question. I typically watch TV 0 to 2 hours a day, 2 to 3 hours a day, 3 to 5, 5 to 10, over 10 hours per day. You can see the response from my last class that the college students responding to this poll say they watch 0 to 2 hours a day. That would be outside of the norm for college students these days, and outside of the norm for most Americans. The average that you can look up is about 4.5 hours a day. Some households will have the television on for 10 to 12 hours, and some high school students reportedly watch 8 hours a day. That's equivalent to a full-time job. So the average American does watch about 4.5, we'll just conservatively say 4 hours a day. Now, I bring this up because if you were to do that for every day, 365 days, that's 1,460 hours per year. And if you divide that by 16, which would be the number of waking hours per day, so if you did nothing else other than watch television, and you slept for 8 hours, so no walks on the beach, no reading books, no going to the library or the movies, you're just watching TV. You've spent 91.25 days out of that year watching television. And if you do that for 50 years, you've spent 12.5 years of your life watching television. That's about average in the United States. Maybe it should come with a Surgeon General's warning that this could take years off your life. I hope it's worth it. But on the other hand, we should consider actually the social impact of this. And it's not benign by any means. Does watching TV influence teenage sexual behavior? Studies have been done which indicate strongly that that's true. Also, does TV influence teenage violence? The answer to that is also yes, unquestionably. And you can look up various studies that have been done on this, and this is also true for video games, violent movies, and so on. Children in TV violence has been particularly well studied, and hundreds of studies on the effects of TV violence on children and teenagers have found that children become immune or numb to the horror of violence that they see on television, but they interpret it as somewhat of a real event nonetheless. And they gradually accept violence then as a way of solving their problems. And they imitate the violence they observe on television, whether that's from realistic type shows or cartoons. And they identify with certain characters, victims, or the victimizers as they appear on television. And these kinds of scenes depicted in this slide are commonplace on run-of-the-mill television. You see them on primetime television. Children of any age can be exposed to this. And in the United States, we think of this as normal. It's normal. And pretty much well accepted. But nearly 2 out of 3 TV programs contain some violence, averaging about 6 violent acts per hour. The average child who watches 2 hours of cartoons a day may see 10,000 violent incidents each year, of which at least 500 pose a high risk to learning and imitating aggression, and the children become desensitized to violence. I think we see the ramifications of this in the newspapers commonly. And I don't mean to be overreactive about this. I think that we can look high and low for reasons for what we see now in our society. And we can make lots of excuses. But these types of facts are irrefutable. And you can say, well, what's the long-term effects? If a child will get over it, right? Well, that doesn't seem to be the case. Here's a study, it was published a while ago in 2003, but it examined the longitudinal relations between children's exposure to TV violence and their aggressive and violent behavior in young adulthood. So this was a long-term study conducted over a couple of decades following children and their behavior as exposed to violence up to about 15 years later, showing a correlation between exposure to television violence and these young adults having a predisposition to committing violent acts. And in order to be in this study, it's not just merely that you had to push a kid in front of the drinking fountain. These are violent acts that would constitute punishable crimes. So this is serious business that has infiltrated really our entire fabric of our society. Violence is throughout our society, and I think that we can see this in the police logs. So this leads me to DNA testing. Forensic DNA testing is nothing like CSI. It's been popularized by television shows like CSI or in movies, glamorized as something that they go out, they find the crime scene, they take the evidence, they go back to the lab, and they look up on the database, and 2 hours later they have their suspect. Well, there's a couple of things wrong with that scenario, and I hope I can kind of inform us tonight what that might be really like. But another thing I would like to tell you is that this looks like there are lots of job opportunities in the field of criminalistic analysis of DNA. That's simply not the case. I mentioned Dr. Carl Ladd and previously as the lead criminalist for the State Connecticut Crime Laboratory. He has a PhD in human genetics. And there are only 2 lead criminalists in Connecticut, and probably a long line of people who would aspire to that position. So you might be really drawn to this subject out of popular interest, but you really need to examine the possibilities for career potential if you're considering this, and be very perseverant if you have that goal in mind. Forensic science is the application of natural science to matters of law. We've been talking in this class about the techniques of biotechnology. DNA as an information containing substance has been applied in this course already. We've seen to agricultural, biotechnology applications, pharmaceutical, medical, cancer biology, etc. In forensic science, we see the application of natural sciences to matters of law. But it's predominantly the same techniques. And I'll discuss that again in the lecture here. Primarily, too, we should remember that this is physical evidence analysis, and as such, it is concerned with the recognition, identification, comparison, individualization, interpretation, and reconstruction of evidence. That's reconstruction of physical evidence, and in this case, we're predominantly concerned with DNA. A criminalist, and I want to compare that to a criminologist, criminalistics is the study and evaluation of the recognition, identification, individualization, and evaluation of physical evidence using the methods of natural science in matters of legal significance, tying those 2 ideas that I just talked about together. Criminology is the study of criminal behavior. So physical evidence examination can link a suspect to a victim. It can link a person to a crime scene. It can link an object to a crime. It can disprove or support witness testimony. And I will talk a little bit briefly later on about the reliability or lack thereof of eyewitness accounts, where now DNA-based evidence has come to bear strongly on either supporting or refuting eyewitness accounts. It can help identify a person. And this is important not only to forensic analysis of crime scenes, but as we'll point out later, natural disasters, war victims, as well as military personnel, and other applications where identification becomes critical. And it can aid in the reconstruction of a crime scene. Remember, I told you before that you leave a part of you behind wherever you go. You're sitting in that seat. Likely, if I really needed to, I could prove that any of you were here today by what you leave behind. You're drinking out of that glass, for example. Some of your cheek cells might slough off into that glass, leaving some of that DNA behind. As a part of physical evidence, then, chain of custody is critical. And the chain of custody begins when evidence is located at the scene, even before it is collected, and does not end until the case has been adjudicated in court and all appeals have been exhausted. Any interruption in this chain of custody can cause difficulties in prosecution of a case. I've already mentioned the 2 state crime lab facilities that have contributed to these lectures, and the forensic science lab services embody these different approaches, including forensic biology, serology, DNA, trace evidence, chemistry, and the instrumentation that's involved with those, as well as identification in terms of latent print selection, which could be fingerprints, footprints, tire marks, etc. Question documents and authenticity, imprints, firearms, tool marks, and forensic photography. But how does DNA forensic testing help in an investigation? Similar to what I was just saying about physical evidence in general, DNA forensic testing provides important linkages, linking a suspect to a victim, a suspect to a scene, or a victim to a scene. All of these provide circumstantial evidence. You might say, oh, really? Yes, circumstantial evidence. However, in the case of DNA testing, albeit strong, circumstantial evidence. With the exception of sexual crimes, which I'll talk about later, DNA places a person at a scene, a person as a victim, or linkages that I previously talked about. And as such, physical forensic evidence is often concerned with patterns. And we're familiar with this from patterns of fingerprints, patterns such as tire marks, the patterns of DNA evidence, but patterns often make up evidence that can be pursued to link a suspect to a crime. In this case, for example, we see this victim and with multiple stab wounds. The patterns. What are the patterns that actually connected a suspect to this victim? Might not be immediately apparent until you notice below that arrow the indentations made by tooth marks. So it's the bite pattern, not the stab wounds in this case, that linked through dental records a suspect to this crime. But patterns. It's patterns that are accumulated as evidence to support a case. And in terms of patterns then, these are the basis for DNA identification similar to fingerprints. You might think of DNA identification then as an exquisite fingerprint that is unique to an individual. So the patterns in DNA markers can link a suspect to a crime. DNA evidence has recently shown to implicate as well as exonerate people in crimes. Notice that the gate on this illustration swings both directions. We have seen people exonerated from death row, from crimes that they could not possibly have committed based on DNA evidence that was later brought to light, even after they had spent, in some cases, decades in prison. So we've seen people implicated by DNA that was left at a crime scene that could not have been solved in any other way before this technology was invented. So DNA evidence has become a powerful tool for shedding light on criminal investigations. If you were to flip a coin 6 times and get heads on all 6 flips, what's the probability of getting heads on the next toss? 1 out of 2, 1 out of 100, 1 out of 1,000, 1 out of 10,000, 1 in a million. You know, keep in mind that the coin that you flipped 6 times, you got heads on all 6. Well, I hope that you've had some statistics or some common sense before you answered this question, because it's always 1 out of 2. And unless you get this question right, I would suggest you avoid Foxwoods. And actually, the lottery is probably a tax for people who don't understand math. But that aside, this is a probability question. And probability comes to bear on how forensic analysis is conducted. I'll get back to this later, but luckily, most of the students in my class got this right. First, a little history by way of this report on the murder at Rodman Dam in 1988. In July 1987, Randall Scott Jones and Chris Riesch, both in their teens, went target shooting with a .30-30 hunting rifle at the Rodman recreation area in Florida. While they were shooting, Jones's pickup truck became stuck in a sand pit, and a fisherman suggested that they ask a couple in a pickup nearby for their help, and Jones and Riesch approached the truck, where Kelly Lynn Perry and her fiancé, Matthew Brock, were sleeping. The two men debated about whether or not to ask them for assistance. The following morning, fishermen found the bodies of Perry and Brock in the woods adjacent to the recreation area. Police investigation revealed that they'd been shot with a .30 caliber bullet, and Perry had been sexually assaulted. Their pickup was reported stolen. In August of that year, Jones was arrested in Mississippi, found driving Brock's pickup. Riesch was arrested the next day in Florida after Jones told police that they were together that night in July. Both were indicted on counts of first-degree assault and sexual battery. A semen sample, we'll just label that EVS, retrieved from Perry's body, and blood samples from Riesch, we'll call that S1, and Jones S2, were compared at a laboratory that specialized in DNA testing. Remember, this is 1988. The resulting DNA evidence indicated which man was guilty of rape. As shown in this diagram, we can see Chris Riesch, S1, has the pattern here shown in magenta, and Randall Jones in blue off to the far right compares with the semen sample from the victim in yellow. So you can quickly identify that the pattern of DNA evidence in this result shows that Randall Jones was the perpetrator of the rape. Using these DNA results and other evidence that the police were able to put together, officials identified Jones as the rapist and were able to piece together the rest of the events of the crime. Without waking the couple in the pickup, Jones shot both Perry and Brock in the head at close range. He and Rhode then dragged the bodies into the woods nearby. They towed Jones's truck with Brock's pickup and left with both trucks. Later, Jones returned to the crime scene, moved the bodies further into the woods, and raped Perry. A representative from the DNA lab testified the chance of having another person having the same DNA fingerprint as Jones was 1 in 9,390,000,000, about twice the Earth's population at the time. After deliberating for only 15 minutes, the jury convicted Jones of murder and rape. The judge sentenced him to a double death sentence, making this the first case involving DNA evidence in U.S. legal history, in which the death sentence was handed down. Rhode was sentenced to 6 years in prison and 20 years probation. So from that point, we have seen the not only the utilization of DNA evidence in crime investigations, but the acceleration of the techniques used to collect and evaluate DNA evidence. So the uses of DNA evidence and DNA analysis now is not only criminal investigations, but most of the cases that are prosecuted in Connecticut and Rhode Island involve paternity cases. Let's face it, these days it's not who says what, it's pretty clear. Genetic disease diagnosis we've already covered in previous lectures. DNA is also used to identify endangered animals, remains from war, and identifying accident victims. But for right now, I want to focus on the top two. So in cases where identification can be accomplished by no other means, DNA has proved invaluable. Whether this is victims of natural disasters or terrorist attacks, such as what we saw in 9/11, or victims of war. All military personnel have a sample of their DNA deposited upon their activities. As we know from previous lectures, but it's worth restating here, DNA is the hereditary material of all living organisms, and it's found predominantly in the cell nucleus. However, I want to remind you that there is DNA associated with mitochondria, which are maternally inherited, and that DNA can also be used in forensic investigations. Nuclear DNA, that is the DNA in the nucleus, is organized into chromosomes. Humans have 46 different chromosomes, 23 of which are maternal, 23 paternal. And these are associated with the standard DNA double helix described by Watson and Crick in 1953. Remember, too, that every time a cell divides, it makes an exact copy of the DNA. Therefore, the DNA is virtually the same in every cell in your body. So if you leave one of your cells behind, it is an identification of you, and it is unique to you. While 99.9% of human DNA is the same, it's that 0.1% that will identify you. And we can look at how that is done. Forensic DNA identification has these basic principles: that each one of us is genetically unique, and that if enough genetic variation were tested, each one of us could be uniquely identified. And as I just mentioned, DNA is found in all cells: blood, semen, hair, virtually any cell with a nucleus. Now, red blood cells do not have nuclei, but there are plenty of other cells in blood that do, white blood cells, for example. So in a drop of blood, there is still plenty of DNA in which to conduct an investigation. So that DNA from an evidentiary sample can be matched with DNA from a suspect to either implicate or exonerate. In terms of DNA casework, forensic analysis, that is criminal analysis, is conducted in over 132 laboratories in 49 states. This is probably up from this data. A large portion of these are comprised of privately run laboratories, and the laboratory protocols are prescribed. None of these labs are doing anything other than cookbook analysis. And you can sort of understand the obvious need for quality control here. These labs conduct over 40,000 cases per year, and there is in every state a large backlog to investigations, unless a particular crime would rise to the top of that list. Many states have a backlog of over 1,000 cases. 80% of these are sexual assaults. I'll get back to this point later. 30% of the time, the suspect is excluded by DNA. That's a good thing. There are over 300,000 paternity cases per year. What are the sources of biological evidence that could constitute forensic DNA analysis? I've already mentioned some of them, but blood, semen, saliva, hair, urine, teeth, bone, tissue, any of these contain cells which DNA could be derived from. But these sources could be recovered from various different items, and you could think about where you leave your DNA behind. Cigarette butts, cheek cells sloughing off, and we will do a project in the last lecture where we isolate our own DNA from your cheek cells, so I can show you exactly how that's done. But cigarette butts, a small number of cells could be left behind there. And we should keep in mind here that those could be recovered directly from a crime scene, or a suspect could be investigated, and those could be recovered from an interview. Gloves, obviously, skin cells, bandanas, ski masks, baseball caps, general clothing, condoms, inside and out, stains on furniture, pillows, sheets. The armchair that you're touching right there now is probably leaving some DNA behind. Hair clips, lipstick, of course, letters, envelopes, stamps. Plant and animal sources can also be evidence. I'll point out some cases that I am aware of where plant material associated with a crime scene could link a suspect to a particular area. Things that you ate for dinner, whether that's broccoli that came from a particular restaurant, could be genotyped, as well as animal sources. Pets leave their hair behind, whether that's on a suspect or a victim. All of these involve analysis by PCR. You'll remember this from a previous lecture, but if you're just joining us for the first time in this lecture, let's remember what PCR is really all about. That stands for the polymerase chain reaction, and its products are analyzed using gel electrophoresis. Gel electrophoresis is the separation of molecules such as DNA, RNA, and proteins based on their charge and their size. Electro refers to the energy of electricity that's used to separate molecules on the basis of their charge, and phoresis means to carry across, from the Greek verb phoros. And so a gel is a matrix in which we can apply an electric field to separate molecules as they run through that matrix on the basis of their size towards a particular pole set up by an electrical current. We should also recognize some of the tools of the trade, so to speak, in molecular biology. For example, the micropipetter, shown in this slide but visualized here, was invented by Walter Gilson, and you can look up his name from the University of Wisconsin. And it's likely that molecular biology in general would not have occurred without the invention of this instrument. And most molecular biologists that I know are born with one of these in their hands, and it's hard to pry it from their fingers. This is one of the tools of the trade. Even with today's robotics, still today, the micropipetter is important for all molecular biology, including the applications that we discuss here. The reason is most of these reactions occur in very small volumes. So this instrument was invented to reproducibly measure microliter quantities of liquids reproducibly. So from 0.1 microliters up to a milliliter volumes, these instruments can reproduce those volumes. And typically these are fitted with very small pipette tips that are disposable. And the small tubes that are shown in this slide typically are the types of test tubes that are used in molecular biology for sample collection and storage. So certainly the Eppendorf tube, commonly called, or the Gilson, we call it a pipetteman, are stock and trade for the molecular biologists, but also for forensic applications. So gel electrophoresis. What is gel electrophoresis? Well, what is a gel? Typically, this matrix, a gel, is made of agarose for most applications, but you can see gels also made out of polyacrylamide, starch, and other matrices. But typically, gels that are made from agarose are purchased from supply companies, stored dry like this, and then weighed out in known amounts, dissolved into a liquid, most usually water, shown in the slide here in this flask. But you weigh out an amount so that you have a known matrix consistency, and the volume of liquid that's added, whether that's buffer or water, also makes a constant matrix. And then it's heated, typically in this very fancy laboratory instrument that if you buy it from a supply house will cost twice as much as if you bought it from Walmart, but it's usually microwaved into a molten liquid that's then poured into one of these molds like this, and we add a comb that we can load then samples after the matrix has solidified. The comb is removed, making wells into which the sample can then be loaded. The gel then is loaded into a gel box, such as this, in which the comb is removed and a buffer then is added to this matrix. And the lid is closed, so you can't put your fingers into it once electricity is being applied. A positive electrode is applied to one side, a negative electrode to another side through a power source, which you can regulate both the wattage and the current. So that's the matrix. The samples then, I should say, that agarose is a long chain of sugar molecules, and as a polymer, it's usually derived from algae and used in this type of horizontal electrophoresis unit. Gels are loaded by taking the sample from this Eppendorf tube with the micropipetter, or the pipetteman, and loading the sample into the wells of the gel. Typically, a dye is used, shown here as a blue dye, because many of these solutions are clear, so that a clear solution into a clear gel, and you really can't see what you're doing very well. And that dye typically allows the solution to sink into the well. So the sample then is loaded into the gel, the electric current is applied, and we can adjust this, and the adjustment then of the current determines how quickly that sample will migrate through the gel. And then the DNA can be stained with a dye or a fluorochrome that will allow it to be visualized under UV light, shown in the center here, under a UV light box that can be photographed, and the data can be collected this way. So that large molecules run very slowly through the matrix, small molecules run very quickly, and therefore over time in an electric current they can be separated and visualized. We can also add a molecular weight marker to the gel, that's to one side, so that we can calibrate it and see what known sizes of DNA fragments are generated. So the applications of gel electrophoresis are DNA fingerprinting associated with the polymerase chain reaction. And this has become very valuable because you can't see one molecule of DNA in gel electrophoresis. But if we amplify that one molecule many times through the application of PCR, we can visualize it very quickly using gel electrophoresis. And it's the same technique that has been used in recombinant DNA technologies that have been described previously in this class, and here applied to forensics. But we see that these same technologies have been applied to cancer biology, vaccine development, all of the other different technologies that we've described. Keep in mind that DNA carries a net negative charge, deoxyribonucleic acid. So as an acid, it has negative charges associated with the phosphate groups that form the sugar phosphate backbone of DNA. Therefore, carrying a net negative charge, it will move in the electric field towards the positive pole. Every student once in a while will have the poles reversed, and they will do that hopefully only once, because the samples then will migrate off the gel in the opposite direction, losing their sample. However, DNA will migrate towards the positive pole over time. So, as I mentioned and shown in the diagram here, large DNAs will migrate very slowly through the matrix as they work their way through. Smaller DNAs will move more quickly. And if you leave this going too long, of course, they will migrate right off of the gel. And every student will do that about once also. If you have the current turned way down, you can leave these going overnight. And sometimes this is advantageous because if you run the gel very slowly, you will increase the resolution of the separation. So as separation continues, as the process continues, the separation between fragments increases. And then, as I mentioned, these molecular weight markers, which can be purchased of known molecular weight, can be applied to one of the lanes as a standard, and you can compare directly the size, the molecular weight size. So you can actually determine mathematically then how many base pairs of a given fragment of DNA are associated with that fragment by evaluating its migration to these known molecular weight standards. And this can be done highly accurately. So what are some of the DNA technologies used in forensic investigation? I have mentioned PCR analysis, and I will get into that again in detail in just a minute. But that is not the only technology that's applied for forensic investigation. Prior to the utilization of PCR, the digestion of DNA by restriction enzymes, these are enzymes that recognize specific sites in DNA, can be applied, and that different enzymes can be used to identify individually specific fragments from a sample, and these have been called restriction fragment length polymorphisms. In other words, if I take DNA sample from you and apply a restriction enzyme to it in a DNA sample from you, these will have different lengths according to their individual restriction sites. So we can actually identify people according to their polymorphisms, the differences in DNA that relate to where these enzymes digest that DNA. However, the probability of two individuals having the same RFLP is not as great as it is for PCR. I will talk in a minute also about STR analysis. This is short tandem repeats, and I will relate this to how this is used in connection with PCR to identify people with a resolution that is greater than that of restriction fragment length polymorphisms. Mitochondrial DNA analysis is also used owing to the fact that sometimes mitochondrial DNA is more resistant to being broken down in a sample. And those markers, however limited, can be used to connect a suspect to a crime, etc. Y chromosome analysis is used in some paternity cases, although there are other markers that are more useful. But Y chromosome analysis is also proven useful for certain anthropological studies. I will leave it up to you to read the more detailed descriptions of what RFLPs are here, but this is a technique, again, used for analyzing the variable lengths of DNA fragments that result from enzymatic digestion. PCR, though, the polymerase chain reaction, is used to make millions of exact copies of DNA, and this is predominantly the technique that's used in forensic analysis today. This is in conjunction with analysis for what's called short tandem repeats. You find throughout the human genome repeats of short regions of DNA that can be used to evaluate specific regions or loci within nuclear DNA. It's these specific short tandem repeats that are used in forensic analysis. I want to clarify that when forensic analysis is conducted, we are not searching for genes, that would be coding regions for proteins, but rather anomalies associated with short tandem repeats. And the FBI uses a standard set of 13 of these short tandem repeats that occur throughout different chromosomes in the human, represented for the CODIS, which is a software program which operates locally, statewide, and national databases. Mitochondrial DNA is associated with the small number of genes, that would be less than 450 genes, or DNA in non-coding DNA in mitochondria. And as I mentioned, the Y chromosome, which is important as it is passed directly from father to son, may link familial DNA to certain cases. In some cases, for example, have been adjudicated based on DNA analysis, not of the suspect directly, but it was found out that it was either a father or another relative with the same Y chromosome. So the polymerase chain reaction. We need to understand the basis of this to understand how it's used forensically. So if you have a given DNA sample, I want to go over how the PCR reaction works to amplify any piece of DNA. I want to emphasize that this technique is extremely reliable and extremely sensitive. So, given an original target of DNA shown here as double-stranded up in the upper left of this slide, the first step would involve denaturing that DNA by heat into single strands. So separation of the strands is the first step. The next step is to use primers as targets for known sequences. In other words, you have to know some part of a sequence in order for this reaction to work. Next, the extension of those primers is accomplished through a polymerase that will bring in the appropriate A's, T's, G's, and C's that will match the complementary strand of that single strand. The reason that the primer is required is that polymerase will only function on a section of double-stranded DNA. Therefore, we make this first double-strandedness by the location of those designated primers. I'm just giving you this now as an introduction. I will go over this in more detail in a moment. But then after those, that is extended through its complementarity, the heating then reoccurs, and that double-stranded copy is separated again into two other unique strands of DNA, and the copying then continues. So to go over this diagrammatically, DNA to be amplified could be collected from any of those samples that I previously mentioned. Cheek cells from a beer bottle, bandana, swipes, inside of an envelope. So DNA to be amplified is first collected. And there are millions of different genes or sequences in any DNA sample. Now you have to remember here in a forensic sample that the sample may have been subjected to less than perfect conditions, say a drop of blood on a sidewalk, exposed to heat, let alone that if a dog licked that sample. And typically we don't have the luxury, if you will, of going back for a second sample. So sample protection and chain of custody is very important at this phase. Primer sites have to be chosen. I already mentioned that in this analysis we're interested in short tandem repeats. Therefore, these are known primer sites. If I were interested in amplifying a known gene, say your hemoglobin gene, we could recognize primer sites associated with that sequence and amplify that. In this case, the CODIS of 13 different loci that are recognized by the FBI for forensic analysis constitute known primer sites. So these are known regions of DNA. So the primers then are well known. The things that are needed for amplification then are the primers for these 13 different loci, the DNA polymerase, which in the PCR reaction must be heat stable. You'll remember from the inventor, Kary Mullis, that this came from microorganisms that were recovered from the hot springs at Yellowstone National Park, where these organisms are capable of replicating their DNA in boiling water. I guarantee you that if I put you in boiling water, you're not going to feel like replicating your DNA, but these organisms are capable of doing this, and this polymerase will function then at high temperatures, and the DNA to be amplified. So without a short piece of DNA or RNA to attach to, DNA polymerase cannot copy. Therefore, the primers then are essential, and their recognition to create a double-stranded region, which is then recognized by the DNA polymerase, which then begins to synthesize the complementary strand on that single-stranded DNA. We also need the base pairs, the adenine, thymine, cytosine, and guanine of DNA need to be present. PCR requires several cycles of amplification, where each cycle consists of 3 temperature changes. First, starting the cycle depends on a starting temperature, which would be about 94, 95 degrees C, which separates DNA into single-strandedness. A lower temperature, 50 to 60 degrees C, allows the primers then to bind, recognizing their targets. A slightly higher temperature, 72 degrees, allows the DNA polymerase to attach to the primers and copy the DNA strands. So let's go over this. First, the DNA is denatured by heating it at 94 degrees, and this separates the double-stranded DNA into single-strandedness. Next, the temperature is lowered to 54 degrees, and the unattached primers recognize their specific sites, and they anneal. Then, in the presence of A's, T's, G's, and C's, we make 2 new DNA molecules directly from that template. Copying is completed for each strand at 72 degrees, and then the process is repeated. So now those 2 copies are then again separated and copied again, and therefore we can go from 1 molecule into billions rather quickly by the repetition of that thermocycling. So I had mentioned already STRs. STRs are used in identity testing. These are short tandem repeat sequences. So if you look at this slide, what is shown in blue is the repetition of a TA, a TA, a TA many times over. Tandem repeat, TA, TA, TA. There are variations that exist throughout the human genome in tandem repeats. So that you, for example, might have a short tandem repeat of, say, for any one loci, that is used forensically, you might have a short tandem repeat of, say, 12 TAs in a row, for example. You might have one that is, say, 24 bases long, and you might have one that is, say, 48 bases long. If I take a primer and situate it at the 3' end of this and at the 5' end of this short tandem repeat, I will generate a very short fragment for you, an intermediate fragment for you, and a longer fragment for you. Therefore, if I run these out in gel electrophoresis, yours will run at the top of the gel at 48 bases, yours will run intermediate at 24, and I will identify yours as the shorter fragment at 12. Therefore, based on the variation that exists naturally in the human genome, I can isolate differences in short tandem repeats and use those for identification. You follow? So the CODIS of 13 core STR loci within the human chromosomal positions are shown here. They are throughout the different chromosomes represented by their identification markers on this slide. The process of copying DNA strands is repeated 32 to 35 times through PCR, and we can go then from one molecule of DNA, which you cannot see on a gel, out to many copies which can be stained and visually identified as a very large fragment which can be stained and visualized within a few hours. And as you can see, this equipment does not take up very much space, and in fact can be in a mobile unit. So over 1 million copies are generated through the 32 cycles of the PCR chain reaction, and these copies then can be easily detected in gel electrophoresis according to their size. So as I said, they will migrate towards the positive pole of the gel, separating them by their size then. I've already used the word CODIS. What is a CODIS? A CODIS is the combined DNA index system. And these 13 markers are the same, used in all 50 states to identify victims or suspects based on their DNA profile. The PCR copies DNA exponentially through multiple thermal cycles. And to do this now, fluorescent labels can be added for amplification of DNA from a very small sample. As I've already said, you can't go back and get another sample from either perhaps a suspect or a victim. So the utility of this depends on accuracy of detection, as well as being able to multiplex these DNA samples according to the STRs and the different fluorescent labels. This is what the laboratory PCR instrument looks like. An instrument like this would fit on a desktop, and the input is DNA, your sample, the PCR enzymes, the primers, the individual nucleotides, and perhaps the fluorescent label. The block that's shown there is a heating block which will go through those thermocycles accurately and timely. And as you can see with the computer keyboard shown in the left there, that this can be programmed. And in our laboratory, we use this for doing PCR in individual samples, but for the CODIS of markers, this is prescribed. The output then would be the specific DNA fragments that can be visualized, and the sizes will vary according to the variation of individuals associated with those STRs. And it's this multiplexing of PCR which allows the analysis simultaneously of different markers, with sensitivities down to lower than 1 nanogram of DNA. And these, I mentioned the different laboratories which conduct these types of analysis. These are made as kits that make it easy for labs to just add DNA samples to a pre-made mix, which limits the QC that needs to be conducted on these, and there's very little variation, hopefully, lab to lab. So a defense then is no longer really seriously taken that, oh, it was a laboratory screw-up. Those are so low as to not be really part of a worthwhile defense at this point. So let's look at what this might look like. In terms of identity testing for PCR, let's just say using 4 rather than 13, just for simplification purposes in this diagram. But you might have size standards in one lane of a gel, followed by victim's DNA, suspect 1's blood, 2, 3, and an evidentiary sample, and then another size standard shown to the far right. What would be the conclusions? I mentioned that this is natural science, and as a science experiment, the possible conclusions from running this analysis as an experiment are that suspect 1's DNA was at the scene, suspect 2's DNA was at the scene, suspect 3's DNA was at the scene. None of the DNA was at the scene. Notice it doesn't say the person was at the scene, but rather the DNA was at the scene. Or that multiple suspects' DNA was at the scene, or lastly, that the data was inconclusive. These are the possible experimental results. So if we were to run this sample shown in the wells in this gel, that these are the possible results with the markers shown off to the left and the far right. And the victim's DNA compares with the evidence from the 3 suspects, and you can see a complete match then between the evidentiary sample and that of suspect number 1. Let's look at what real data might look like. This is a case study showing evidence from the state versus Michael DeCorso, where there was a homicide and no DNA was recovered. There was rape evidence where DNA and semen samples was recovered from 2 teenage female victims. In this analysis, for the marker D10S28, we show the DNA marker ladders to the far left and the far right and in lanes in the middle. And the control sample after the markers indicates that the PCR reaction was working as a control, as a positive control. The next lane you have the victim, which shows a single fragment, and the defendant, Michael DeCorso, with 2 distinct fragments. And then on the other side of that molecular marker, we have evidence 1, 2, and 3, and we can see bands corresponding to the same size as Michael DeCorso's DNA in evidence 2 and 3 from the 2 teenage rape victims. So those size fragments correspond to the size fragments of Michael DeCorso. The population frequency of that marker, D10S28, is approximately 1 in 50. So if you're the defense and you see this evidence, you say it wasn't my guy. It perhaps was the 51st guy. What have you demonstrated here? So let's look at another marker. D4S139. We see again that the control DNA generates a fragment indicating that the PCR reaction was functioning. The victim's DNA separates into two fragments, as does Michael DeCorso's, but they're of different size than the victim. We see that evidence 2 and 3 from the two rape victims again match those of Michael DeCorso. Note that the bands of evidence number 2 are relatively faint. This still scores as a positive. Remember, we're interested in size, not intensity. The size of the fragment here correlates precisely with the DNA from Michael DeCorso. And in this case, D4S139, the population frequency of the defendant's genotype is 1 in 90. Somewhat better, but again, if you're the defense, you might argue it's the 91st guy, not my guy. So let's do another one. D5S110 generates a fragment, a single fragment from Michael DeCorso, also corresponding with that of both of the teenage rape victims. That one occurs 1 in 10. Let's do another one, TH01. That generates two fragments from Michael DeCorso that again correspond to the teenage rape victim's DNA with a frequency of 1 in 70. So I ask you now, what is the probability of having all 4 of these markers corresponding to Michael DeCorso's DNA? Well, previously I asked you about the probability of flipping a coin, and you know now from probabilities that you multiply these together. So 1 in 50 by 1 in 90 by 1 in 10 by 1 in 70 gives you a 1 in 3,150,000 probability chance that anyone would have those same 4 in a row, and that was enough to prosecute Michael DeCorso for that crime. Now we have 13 markers, and the probability of getting 13 markers now for any one person is more than all of the people that have ever been born in the history of the earth. So the chances of scoring for all 13 markers exceeds that of all the people that have ever been born, and that would be unique to one individual. So in order to be prosecuted then, you need to score 13 out of 13. If you score 12 out of 13, that's not good enough. And chances are they'll look closely at your relatives. But forensic applications now have been in place for many years. You might ask what went wrong with the O.J. Simpson trial if you're familiar with that history. And I invite you to read about it. I won't go into that right now. But certainly they took days to explain the PCR reaction famously to the jury. Days? I just took several minutes, and I hope I did a better job than either the defense or the prosecution did in that trial. If not, then you might want to review what I just talked about. There's been a dramatic growth in DNA-based forensics, but as I mentioned previously, that doesn't translate into very many job opportunities. Paternity testing still is the largest application on not violent crimes. I also mentioned the use of non-human DNA. Here's an example where a perfect DNA match from cat short tandem repeats was used to implicate a suspect in a crime where there was a homicide in 1994 and a plastic bag was found in a shallow grave with a bloody jacket and trace hairs. The hairs belonged to the suspect's cat, Snowball, which was identified using short tandem repeats. There was also this example of dog DNA, which contributed to murder convictions where there was a double murder and 2 suspects in Seattle in 1996. A couple had been tortured and shot along with their pet dog. The blood on the suspect's clothing was found to match that of the dog, with a 1 in 3 billion match probability in random canine populations. I am also aware of a case which was prosecuted in Las Vegas where there were several violent sexual assaults which occurred in the backyards of suburban houses. The only evidence linking a suspect to the crime were jeans, literally pants, found in the suspect's closet with grass stains. DNA from the grass stains was removed and found to correlate to the lawns in the houses where those crimes were perpetrated. You might say, well, how unique are grass stains? Well, it turns out that unique enough to find populations of grass that correlates with different housing projects. A housing project might buy a lot of grass from this supplier and another one from another supplier, in which the short tandem repeats or the RFLPs from that grass population could be correlated in that case to individual grass stains. Or I'm aware of another case in which a crime was committed in a desert and that a certain plant material was found associated with the truck in that crime that correlated only to that particular site in the desert. So the point is that DNA does not have to come only from humans. Even samples, as I mentioned previously, from where somebody ate previously could be analyzed and correlated, say, to an individual restaurant. So grass stains linked to lawns, vegetables, DNA linked to restaurants, rare or unusual plants linked to vehicles, marijuana samples found on suspects or in their car or their house could also be connected to a crime scene. Since Connecticut started a DNA felon database in 2004, it has been collecting DNA from felons, and currently, how many felons would you estimate were on the Connecticut DNA felon database? Would that be 1 in 50 males, 1 in 500, 1 in 1,000, 1 in 10,000? None of these answers is correct. Well, actually, that'd be 1 in 50 males. So that's be kind of surprising maybe to some of you, but this is an all-felon database, and I want to get into this aspect of this lecture on the second part as it relates to DNA databasing and the debates that arise about this type of information. Let's look at another case study in terms of how DNA evidence could be used to solve a crime. In this case study involves Rene Pellegrino. This is a local case and involving the murder of Rene Pellegrino, whose body was found on June 25th, 1997. Rene Pellegrino was an accomplished scholar with a law degree who had become addicted to crack cocaine and turned to prostitution in order to support that habit. She was 40 years old and pregnant when her naked body was discovered in a cul-de-sac off of Waterford Parkway South in Connecticut, near New London. She herself had just been released from a 3-week prison stay when she was apparently picked up by her killer in downtown New London on June 25th, 1997, and murdered and left naked in the cul-de-sac. She'd been strangled, and her killer had left her body in what the judge described later as an extreme manner. The case went cold. No suspects were arrested for 13 years. On June 1st, 2010, the police in New London made an arrest in the Pellegrino case, and they charged Dickie E. Anderson Jr., 40 at the time, with murder in the 40-year-old Pellegrino's death. The result was, the arrest was the result of the cold case investigation that went 13 years into the investigation of Pellegrino's death. Anderson, who would have been 27 at the time of the crime, was arrested as a result of the cold case investigation conducted by the Southeastern Connecticut Cold Case Unit. The arrest warrant was sealed. So this photograph of Dickie Anderson was taken at the New London County Courthouse upon his arrest from the June 1st issue of the New London Day. The police used a combination of evidence to build the case against the man accused in the 1997 murder of Rene Pellegrino, including DNA evidence. But they also included inconsistent statements made by Dickie Anderson to police over those years in order to piece together the evidence that led to his arrest, as well as his own admission that he had been with Rene Pellegrino shortly before her body was discovered in Waterford that morning. Over 13 years then, this case, including DNA evidence, comprised what was used to make the arrest. If you had to vote right now, based on what I told you about this case, would you say that Dickie Anderson was guilty, not guilty, or I can't vote that I don't have the facts of the case? Can I just see a show of hands? Guilty? Not guilty? Don't have enough evidence. It's not a trick question, actually, but this is the common question that's asked during jury selection, and many people are tempted to respond incorrectly that they don't have enough evidence. The correct answer would be not guilty in the United States until proven otherwise. In a second case, police charged Anderson with killing 29-year-old Michelle Comeau of Norwich in 1998. Anderson had been, as I mentioned, previously charged with the murder of Rene Pellegrino, and the Comeau case did not involve DNA. I'll get back to this in a minute. Both of the women had been working as prostitutes and were victims of strangulation. Police also found Comeau's body dumped along an access road to Norwich in the industrial park near Dodd Stadium in May of 1998. Anderson had acknowledged that he knew, in fact, both victims. He told police that he had been with Pellegrino on the night that she disappeared from downtown New London. Pellegrino had been strangled, and her killer had posed her naked body in the form of a crucifix. In May 1998, police found Comeau's body dumped along an access road to the industrial park she too had been strangled, and the state had claimed that her killer was in the process of posing her body in a similar manner, but was interrupted and left the scene quickly. Anderson's DNA was found on Pellegrino's body, but there was no DNA evidence to link him to the Comeau case. He admitted to knowing both women, who were frequently visitors of Dickie Anderson Sr.'s house. And Dickie Anderson admitted to having sex with them on the day that they were killed. One inmate had reported that Anderson admitted to killing Renee and said several times that he would never have done that had he known she was pregnant. She was 17 weeks pregnant when she was killed. The police also spoke with former girlfriends of Anderson who told them that he had had rough sex with them in the past. One woman said that Anderson had threatened her and said that he had gotten away with killing somebody. The woman said Anderson said he fought with a prostitute who kept asking him for money, and that he had hit and killed the girl in Bates Woods in New London. The police also interviewed a girlfriend who broke up with Anderson in 2000. She recalled that twice Anderson choked her so hard he left red marks on her neck. She turned over to police a picture of her injuries that she said that a friend had taken. Another former girlfriend, who Anderson was convicted of strangling in 2008, said the two had argued about her getting a job and that Anderson threw her to the floor and began choking her. She said if police did not break into the apartment and physically remove Anderson from her, she thinks she would have died. Dickie Anderson Jr., a self-confessed trick artist who told police he had often traded crack cocaine for sex with prostitutes, was connected by witnesses to both of the women he was accused of killing. In 2008, the State Forensic Laboratory had identified police that DNA taken from Pellegrino's body matched a DNA sample taken from that of Dickie Anderson. The laboratory also found DNA on Pellegrino from an unknown source. Anderson's previous convictions included the following list: 2007, third-degree assault; 2005, violation of a protective order; 2005, September, second-degree failure to appear in court, second-degree criminal mischief; 2003, violation of probation, evading responsibility, third-degree assault; July 2002, interfering with a police officer, 1999, second-degree assault. So, Dickie Anderson, if you had to vote now, based on what you've seen, guilty, not guilty, can't vote, don't have enough evidence, can I see a raise of hands again? Guilty? Not guilty? Not enough evidence? Well, the jury convicted Anderson of one of the two murders, that of Renee Pellegrino. A mistrial was declared in deadlock over the second prostitute's murder, owing actually to a lack of DNA evidence, which is why I bring up this case in particular in this class. What about the death penalty? This is a dilemma that our entire country grapples with and is divided on now, as many people probably in this room might be. For or against. Here are the states with and without the death penalty now. This might be divided in your mind along ethical, spiritual, or religious reasons. But certainly you can see how this plays out. Recently, the state of Connecticut in 2012 repealed its death penalty on the heels of a trial which took place over a brutal murder and house invasion. The death penalty in Rhode Island was abolished in 1852, reinstated in 1872, and then again legally abolished in 1984. In Connecticut, the repeal of capital punishment involved some historic Senate debates. But it was the Cheshire, Connecticut home invasion murders, which occurred on July 23rd, 2007, which really caused some of this back and forth decision. When a mother and her two daughters were murdered during a home invasion, as reported by the Hartford Courant, as possibly the most widely publicized crime in the state's history. In 2010, Stephen Hayes was convicted of the murders and sentenced to death. His accomplice Joshua Kamaszewski, was also found guilty on October 13, 2011, and sentenced to death on January 27, 2012. This story starts with the late afternoon of July 22, 2007, when Jennifer Pettit and her daughter Michaela went to a local grocery store in Cheshire. They picked up food for an evening meal, which would be prepared by Michaela. They, along with Jennifer's other daughter, Haley, would be killed several hours later in that home invasion. I've compiled the story of that home invasion, which I will leave up to you to read, but it involves the home invasion and basically a story gone awry with the plans of Hayes and Kamaszewski, where they had planned a home invasion and a robbery in which they beat William Pettit, MD, with a baseball bat, tied him up in the basement, and then discovered that their take in the crime would not be that great had they just absconded with what they had. And Hayes convinced Jennifer Pettit to withdraw $15,000 from a line of credit from a local bank. The gas station's video surveillance shows Hayes putting $10 worth of gasoline into 2 cans he had taken from the Pettit home, and after returning to the house, unloading the gas. He took her to the bank. The bank surveillance cameras captured the transaction showing Jennifer Pettit, where she informed the teller of her situation. She indicated that the assailants were being nice and that she believed they only wanted money. The teller called 911. During the time after they returned, however, the home invasion escalated with the aggravated nature of their crimes, and Joshua Kamaszewski sexually assaulted the 11-year-old daughter and photographed the sexual assault of the youth on his cell phone. He then provoked Hayes to rape Jennifer Hawk-Pettit. While Hayes was raping Hawk-Pettit on the floor of the living room, Komischevsky entered the room announcing that William Pettit had escaped. Hayes then strangled Hawk-Pettit, doused her lifeless body in parts of the house, including the daughters' rooms, with gasoline. The daughters, while tied to their beds, had both been doused with gasoline, and each had her head covered with a pillowcase. A fire then was ignited, and both men left the scene. 17-year-old Haley and 11-year-old Michaela both died from smoke inhalation. William Pettit was able to free himself and escape, and eventually testified in court. The whole invasion lasted about 7 hours. The scenario was actually revealed in a confession by Stephen Hayes hours after the killings, and police detected a strong stench of gasoline throughout the interrogation. Each perpetrator blamed and implicated the other in the aggravation and the acceleration of the crime. They even tried to implicate the father. Joshua Komischevsky is now on death row, serving a death sentence. Stephen Hayes would like to commit suicide and is also on death row, convicted on 16 counts, sentenced to death on 6 counts of capital felony. The death penalty. Do they deserve it? Is it a deterrent? Is it humane? Is it inhumane? These are difficult questions. For or against? Our country is still divided. We are the hollow men. We are the stuffed men, leaning together, headpiece filled with straw. Alas, our dried voices, when we whisper together, are quiet and meaningless, as wind and dry grass or rats' feet over broken glass in our dried cellar. Shape without form, shade without color, paralyzed force, gesture without motion. Those who have crossed with direct eyes to death's other kingdom, remember us, if at all, not as lost violent souls, but only as the hollow men, the stuffed men. T.S. Eliot.